Welcome. Today we will be conducting a discussion on the ELISA disease detection model by modeling the general questions and components associated with antigen and antibody detection of the SARS-CoV-2 virus. The antigen detection ELISA model measures the antigen-antibody interaction of an unknown virus and known antibody. Its main goal is to test for the presence of antigen in a patient's sample. This model has 5 primary components, which include the antigen found in the patient's sample, the primary antibody that binds to the antigen, the secondary antibody that binds to the constant region of the primary antibody, an enzyme called horseradish peroxidase that attaches to the secondary antibody and catalyzes a color change, and finally the substrate necessary for binding to the enzyme and inducing the color change to indicate a positive result for having the infection. The antigen detection model setup involves using a plastic microstrip plate with 12 wells total, with each strip having a well for a positive control, negative control, and 2 patient samples. In the model shown here, the test begins by adding the antigens to the designated well that may be potentially SARS-CoV-2 positive in the patient's sample. Then the primary antibodies are added to the well, where the antibody interacts and binds to its specific antigen, the SARS-CoV-2 virus. The sample is then incubated for 5 minutes to ensure strength of antigen-antibody interaction. A wash buffer is additionally added to the well to rinse out any remaining primary antibodies that did not bind to the SARS-CoV-2 antigen. The wash buffer is added an additional time to purify the sample. Once the interaction between the antigen and antibody has been established, enzyme-linked secondary antibodies are added into the well to bind tightly to the primary antibody. The sample is incubated for an additional 5 minutes to enable the interaction between the primary and secondary antibodies to occur. A wash buffer is added to the well to remove any secondary antibodies that did not bind the primary antibody. The process is further repeated 2 more times. In the final step, the substrate is added to the well and incubated for 5 minutes. During this period, the presence of SARS-CoV-2 stimulates the enzyme on the bound secondary antibodies to catalyze a reaction, causing the well to turn blue, which indicates a positive patient sample. As we have found in the antigen detection model, this model has the ability to identify the presence of the SARS-CoV-2 virus in a patient's sample. This model also has the capability to model positive control. In a positive control, the model utilizes similar components as the patient's sample, where it will have the correct primary antibody and secondary antibody combination present that is capable of binding the target antigen. The model follows a similar process to studying a patient's sample, where it is necessary to add the antigens to the designated well. Then the primary antibodies are added to the well, where the antibody interacts and binds to the SARS-CoV-2 virus. The control follows the same steps of incubating for 5 minutes and adding a wash buffer. Once the primary antibody successfully binds its target antigen, enzyme-linked secondary antibodies are added to bind to regions on the primary antibody. The process of incubation and adding a wash buffer to the well is repeated additional times to remove unbound secondary antibodies. In the final step, the substrate is added to the well to stimulate the enzyme on the secondary antibodies to catalyze a reaction necessary for identifying the presence of the SARS-CoV-2 virus. In contrast to a positive control, a negative patient sample will have the incorrect antigens present where no binding between the primary antibody and target antigen occurs. The process begins by showing the addition of antigens to the designated well. Then, as seen before, the primary antibodies are added to the well where it has the ability to interact and bind to its specific antigen. In this particular sample, the antibody is not specific for the circular SARS-CoV-2 antigen and cannot bind to the target antigen. Following the same steps of incubation and adding a wash buffer, the unbound primary antibodies will be washed away. As the sample now shows no presence of antibodies to combat antigen exposure, there will be no further binding of enzyme-linked secondary antibodies or substrate. The lack of antigen-antibody interaction causes the well to remain clear and therefore indicate a negative result. When comparing antigen and antibody concentrations, it is important to identify the length of time post-infection when the antibody finds its target antigen. In the graph shown here, the solid line represents antigen and dotted line represents antibody production. On day 35 of the patient post-infection, we can see here that antigen concentrations are at its lowest and antibody production is at its peak concentration. At this stage in the infection, an antigen detection ELISA would not accurately diagnose the patient due to the concentration of antigen reaching 0, which cannot be easily detected by ELISA and will be unable to offer conclusive results on the patient's sample. And since the antigen detection ELISAs are also able to detect viral proteins, the detection of viral DNA through a viral PCR test would be the most beneficial when the antigen is at its highest concentration. Especially in the graph shown here, day 15 shows the antigen at its peak concentration, which is the point at which the viral proteins are generated the most and will be the easiest to detect through a viral PCR test.
Our question is, could goat anti-rabbit secondary antibodies be used to detect two different types of rabbit antibodies? And we believe that goat anti-rabbit secondary antibodies could be used to detect two different types of rabbit antibodies. This is because our goat secondary antibody, right here, when exposed to rabbit primary antibodies, so we have type 1 here and type 2 here, when the secondary antibody is exposed to either of the primary ones, it'll begin to bind to the constant region of the secondary antibodies. So this region here for type 1 and this region here for type 2. This region of the primary antibodies are unchanging, so they're the same. And because of this, the goat anti-rabbit secondary antibody can be used to detect this constant region and bind to it, regardless of what the type of the primary antibody is. As long as the constant region remains unchanged, our goat anti-rabbit secondary antibodies can be used to detect the two different types of rabbit antibodies. In this scenario, we want to determine whether the same secondary antibodies, in this case goat anti-rabbit, can be used to detect both rabbit anti-influenza antibodies and rabbit anti-HIV antibodies. Here we have our pre-existing influenza primary antibody, and here we have our HIV antigen, which has been recently injected into the rabbit, and the correlating primary antibody to detect that HIV antigen. In this example, it's only the HIV antigen that is present. The influenza antigen is not present, but the influenza primary antibody is. And here we have our secondary antibody. Once again, we want to determine whether the same secondary antibody can detect and bind to both primary antibodies, in this case influenza and HIV. And it can, and this is because the goat anti-rabbit secondary antibodies are only concerned with binding to the constant region of the primary antibodies. They do not care. The secondary antibodies have no binding specificity towards the type of antigen that is present. So regardless of whether it is an influenza antigen or an HIV antigen, as long as the constant region is the same that binds to the correlating antigen, the secondary antibody will bind to both constant regions. Here we want to determine whether we would still get a blue color change when the substrate is added in a direct ELISA. We've previously modeled an indirect ELISA, which you can see on the left, and in an indirect ELISA, all the pieces are there. So we have our antigen present, our bound primary antibody, our bound secondary antibody, and our enzyme and attached substrate. They're all present. And as a result, as we know, this produces a blue color change. In this question, we want to determine if the same blue color change will occur in a direct ELISA. The main difference between an indirect and direct ELISA is that in a direct ELISA, there is no secondary antibody present. Instead, the enzyme is directly attached to the primary antibody, which is directly attached to the antigen. If we were to add our substrate to our enzyme, then we would notice that a blue color change would still occur. And this is because the blue color change is completely dependent on the presence of our substrate and our enzyme. It doesn't matter which one they are located on, which antibody they're located on. It can be the primary antibody, in this case of the direct ELISA, or the secondary antibody, in the case of an indirect ELISA. As long as the enzyme and substrate are present on an antibody, the blue color change will still occur. We know that a blue color change will occur in both an indirect and a direct ELISA. But in this situation, we want to determine whether the signal intensity or the amount of color change will differ between an indirect ELISA and a direct ELISA. In an indirect ELISA, we know that the color change there will be a lot of color change, and the signal intensity will be very strong. But in a direct ELISA, the signal intensity will be weaker, and there will be less of a color change. This is due to the presence of a secondary antibody. The secondary antibody is present in an indirect ELISA, which means the signal will be strong. But in a direct ELISA, there is no secondary antibody present, so the signal will be weaker. The presence of the secondary antibody is very important because while they do not determine whether a color change occurs in the first place, they do aid the primary antibody by helping in detection sorting, purification, and most importantly in this case, signal enhancement of the antigen. Because of this, the presence of the secondary antibody will determine the strength of the signal. And the greater the number of secondary antibodies that are bound to a single primary antibody, whether that be an indirect or direct ELISA, the greater the strength of the signal, and thus the stronger the color change will be. There are a couple advantages of using enzyme-linked secondary antibodies, but one of the main advantages of using these secondary antibodies is that it allows for higher sensitivity of the primary antibody and ultimately higher binding specificity and higher affinity to an antigen. This is because while only one primary antibody can bind to an antigen, multiple secondary antibodies can bind to a single primary antibody. As a result of this, these secondary antibodies work together to enhance the abilities of the primary antibody and allow for the primary antibody to bind to the antigen better and allow for its signaling to become improved.
In an antibody detection ELISA, lab-purified antigen is added to the wells first. In this model, the solid black circles represent the coronavirus S protein antigens. The first step of the antigen detection ELISA allows us to determine whether a desired antigen that would indicate infection is present. The first step of the antibody detection ELISA would allow us to determine whether a patient has antibodies present for a specific antigen introduced to see if they have had previous exposure to the pathogen. After antigen is added, the wells are washed. The wash step is important for rinsing out anything that is not bound to the antigen, and it also prevents non-specific molecules from binding in future steps. Next, the patient sample is added. Patient samples contain a variety of antibodies. If the patient has been infected with coronavirus, the sample will also contain antibodies to the coronavirus S protein. This step is used to determine the presence of SARS-CoV-2 antibodies present in the patient to determine whether a previous infection has occurred, and contains the antigen and the primary antibody. Here, we can see the patient sample has antibodies to coronavirus. An enzyme-linked secondary antibody is added next. In this step, the secondary antibody that has a bound enzyme now attaches to the constant region of the primary antibody. Finally, the substrate is added. If the sample turns blue, which indicates a positive result, the patient's sample contains antibodies to coronavirus. A positive antibody result does not necessarily indicate an ongoing infection, since a high antibody concentration can be present when antigen concentration is low or non-existent. Antigen concentration starts to decrease around the same time antibody concentration increases.
Using our shapes to model the following for an antibody detection of ELISA, we have a positive control, in which we have our target antigen being expressed on both the primary antibody and the secondary antibody, which bind to the antigen and then bind to the constant region of the primary antibody, respectively. By having both these present, we'll get our desired result of the experiment, which would be a positive COVID test result using ELISA. Here is our well, as we know, and this is our positive control. We know this patient will express the necessary antibodies that we're looking for to detect the infection. So moving forward, we'll see that the plastic well will be filled with our purified antigen as it binds to the well. And we see here it being filled, and it binds to the well with its respective purification liquid. So now as we see, we no longer have the liquid, as our washing buffer with detergent washed out all the unbound proteins on the surface of the wells, and now we'll block any remaining proteins on the surface of the wells. So now we'll have our serum antibodies being placed with our purified antigens so that they could be recognized by the specific serum antibodies. And therefore the unbound serum antibodies will then be subsequently washed away, as we will see here in a bit as we pour in our serum antibodies into our plastic well. So we now have our positive control plastic well filled with our purified antigens bound with the specific serum antibodies. Now we'll be taking an enzyme, which is our linked secondary antibody, which is going to be binding to the constant region of our primary antibody. And now we're going to see how our secondary antibody are attached to the enzyme, which will eventually catalyze a color change. So our unbound secondary antibody is going to be like washed away, almost like a second wash, as our enzyme link secondary bond is to the serum antibody. Now that we have our antigen bound with a primary antibody bound to a secondary antibody connected with an enzyme bind, we're going to see how when we add an enzyme substrate, which in this case is going to be TMB, is going to be placed back into that same plastic well. Now the substrate will be causing that color change we discussed, as it'll be in the presence of the enzyme, which will show that we will have this positive result. So we see our enzymes being placed into the well, and it's going to be oxidizing with our substrate. So this is when we get this blue color change, and that is essentially what we're going to be looking for in order to see that we have gotten what we now know as a positive result for our positive control, which is our desired effect. So now that we have our second discussion, which is a negative control. A negative control is essentially a no change or no treatment change with ELISA. So our first control option is having a primary antibody that doesn't bind and will therefore be flushed during the first wash. And this can be seen when we don't actually have any primary antibody, which contains antibodies from the virus. So this could be found when patients actually have no known case of COVID to them, so they don't have any COVID antibodies. And so as we see, our purified antigen won't actually be able to connect to our primary antibody as seen here. And so therefore we won't actually see any connection because there won't be any binding. So during this wash, during our first step with the detergent, you won't actually be able to go into the subsequent step of finding a secondary antibody being introduced to the enzyme or the substrate. And so therefore we can say for our first negative control option, we can simply just have no primary antibody being introduced. So we could use a negative patient sample as a negative control. Now for our second and third options of using a negative control, we discussed if the enzyme or substrate does not in fact bind with our secondary antibody, for instance, the enzyme or substrates that will be able to bind with the constant regions, we can then say that there will be no catalyzed color, which is the change we desire to see if there is going to be a positive COVID exam. So we can say that these could be negative control options as they'll indicate a false positive. And so now we can see that we have our plastic well. Similarly, we'll not have an antibody and an enzyme substrate that isn't going to bind with one another. And as we see here, a bound enzyme bound substrate to the secondary antibody would not happen, indicating a possible false negative if not careful. Now, a negative patient sample will show that a primary antibody did not bind and was flushed originally during the step with the detergent. This is similar to how we were analyzing the original negative control option, so it would be explained in the same way. The sample would not have any antibodies, showing that the patient exposure to ELISA will not have any binding to the secondary antibody. Moving beyond to see if it'll bind to specific serum antibodies, moving on to the enzyme or substrate will not be possible simply as there will be nothing for them to attach to because of the original purified antigen not being able to actually bind to any primary antibody, since there will be none, as the patient will not have any antibodies that will consist of showing that they had COVID. So now for number 11, we discuss how if, according to this graph, could we say use antibody detection, ELISA, to accurately diagnose a patient 30 to 5 days after exposure. So here I'm pointing out where the peak antibodies are on day 30 and where peak antigen or infection have here around day 14. So seeing right here, as these are where the main point of antigen exposure is, we can't concretely say that this is the point where they have had COVID. And we can confirm that they have in fact had COVID due to the actual presence of antibodies. However, due to the fact that they have had a prior infection, ELISA can determine this due to the antigen detection. But we cannot confirm that or in fact diagnose the patient with COVID, since it's now 35 days after exposure, and that the infection period has in fact passed now that we're on day 35.